So this is the conditioned specification. So condition specification is that the ability of the cells to achieve their respective fates by interaction with other cells. Okay, interactions with other cells. So actually here in autonomous specification, there we learn that the one cytoplasmic factor is very important. Okay, one cytoplasmic factor is very important, but here the cytoplasmic factor is not important. Here important the surrounding cells where the cells are or which in which cells they are interacted with the particular cells. That means neighbor cells are very important here in case of conditioned specification. Okay, neighbor cells, because they are totally triggered the particular type of cell to produce the particular type of tissue or cell. Okay, so this is the conditioned specification. If we take one example, in CRG, the 4 cells blastomere is that present in blastomere. So the 4 cells generate normal pluteus larva. 4 cells they generate the normal pluteus larva. But if we separate the 4 cells from the fertilization envelope, each cell develops 1 pluteus larva. Each cell, that means we can see the 4 pluteus larva, where the here in general normal situation the 4 cells they are involved to normal 1 single pluteus larva. But if we separate the 4 cells, then we can see that each cell produce 1 single pluteus larva. That means 4 pluteus larvae are formed. Okay, so this is occurred due to the presence of neighbor cells. That means neighbor cells trigger the particular cell to produce what kind of cells or tissues are needed to produce. So this is the conditioned specification. Another type of specification is there, that is the syncytial specification, and it is seen in the insect. Okay, so syncytial before understanding the syncytial specification, first we learn about what is the syncytial system. That means one cytoplasm present, but there are many nucleus. How is that possible? That means here the nuclear division is occurred. Okay, from one nucleus, many nucleus are formed. Okay, but cytoplasms are not formed, or division of the cytoplasm is not possible here. Only the nuclear division is possible. Cytoplasm here is common. So this is known as the syncytial system, and the specification that occurs for this syncytial nucleus. Okay, so this is known as the syncytial specification, and here we see that one morphogen, that is morphogen, generally diffusible substance, that are very involved here. Morphogen. Okay, just we learn this syncytial specification with a proper example. Okay. Okay, this is an example of a syncytial system. So two substances, one is bicoid and another is the caudal. Bicoid generally present high amount in the anterior portion. Okay, and caudal is present in the high amount posterior portion. Okay, so actually these two are involved for the formation of posterior portion. So two substances are very important for the formation of the different part of the body in Drosophila. In anterior part, where the head is generally formed, the high amount of bicoid but little amount of caudal present. Okay, high amount of bicoid and little amount of caudal present. Then this part of the nuclei are involved. The formation of, not nuclei, nuclei containing cells are involved for the formation of the head. In the second situation, nuclei with the less bicoid and small caudal. That means the cells containing nuclei with very little amount of the bicoid and small caudal, that helps to trigger the cells to form the thorax. Okay. In last one, that means in the case of posterior portion, the nuclei present with the cell and the substance is no bicoid and plenty caudal, that help to produce the abdominal structure. Okay, so these are the actual function of the nuclei. Okay, the nuclei containing cells are very important because nuclei that release these substances, and different part of the cells containing the different nuclei that are involved for the formation of this part, head, thorax and the abdominal structure. So here the syncytial nuclei, these are incorporated into the cells. The nuclei that present within the cells, they generally trigger the cell of that part to form the particular part of the body system. Clear? And this is the syncytial specification. Okay. Here I discuss about the conditioned specification and the syncytial specification. But before that, in my previous video, I explained about the autonomous specification. Okay. And I just explained about the morphogen. What is morphogen? Very little or briefly I described about what is morphogen. This morphogen actually concentration. The concentration morphogen is very important because this concentration trigger actually the cells to develop which part is involved for the formation of the which body part. Okay. So this morphogen are very diffusible and they transport from the source to the whole body part, and where which morphogen concentration where is very important or where is very little important, that dependent on the body structure. Okay. So this is the specification I explained here, the one part of the developmental biology, and you just follow my obviously will follow the, if this is very knowledgeable to you, then you obviously subscribe and share my channel, and I will discuss about another developmental biology topic, the topic of that developmental biology in my next video. Okay, thank you.